I will hand it over to Gary now to begin with the patient panel. Yes, Gary, you're on air.
Yes. Can you hear me? I've changed over to my cell phone. Okay.
Yes,
great.
very well.
Okay, it's fantastic. Dr. June, first, I'd like to just tell you how excited I was, and I think most of the people on this call and anybody who's, who I've talked to about having you on this program. And it's because for a lot of us, especially people with myeloma like myself, in the end, we're going to end up with no new options. And you've provided us with the next best chance after all our options are over, and maybe even better than that, that if we come earlier in the course of the disease.
Yeah.
But I did want to say that earlier when we had a discussion, just briefly, you mentioned something that just struck my heart, and that was that you recently had a reunion with 40 children who were beyond any help, and those 40 children and their parents are with you alive and well. And if anything, I'm sure those parents think of you as a medical messiah. And and at a minimum, your medicine's Einstein. So thank you so much for all that you do.
Well, it was. Thank you. It was that event was. I just can't tell you how. Grateful I am to be able to see this happen. Most people who work hard on a scientific problem or medical problem for a long time, oftentimes don't see the benefit that may happen years later. But to see these children and young adults, it was incredible. And and the good news is they actually look like, from what we can tell, most of them are probably cured. Because we have now sensitive assays for leukemia, and we can't find any leukemia in these patients after the CAR T cells. Now, that doesn't mean they couldn't get leukemia again, but at least from what we can say now is it's going to be a rare event. Most of them, have gone on now, and they just have normal lives. So it's quite remarkable. There is one side effect that's ongoing, and which I should describe, which is, in this case, I mentioned the CD19 molecule that's on these leukemia cells. It's also on normal B cells. And so as long as these patients have these CAR T cells on patrol in them, their normal B cells also get killed. And that turns out that's causes no side effects as long as one gets gamma globulin, which is something often given to people who are going to an area where there's a lot of infectious disease. Gamma globulin is pooled antibodies that the Red Cross makes. And so that's it shows that the CAR T cells are still working, because if we intentionally kill the CAR T cells in the patients, then they start making B cells again. And so in future generations of CAR T cells, we want to make them controllable by the physicians and patients so that we could turn them on or off and then have the B cells come back.
Great.
Yeah.
Well, I can imagine you've made some parents very, very thankful, and certainly made you very much aware of what you've done. So anyway, Dr. June, I thank you again, and thanks for giving terminal patients a new last chance. And my question is, that CD19 happens to be the antigen that you can attack for acute and lymphoblastic leukemia. My question is do you believe, that there is a target on all cancers, that it's just a matter of finding it? And I ask that question because if it's a matter of finding it, then it's just a matter of time. If it doesn't exist, then darn.
Yeah. So I think I can explain in terms of people can understand, because it's really pretty simple. So in the case of T cells, they evolved to kill cells that have new DNA in them. So that means a virally infected cell, for instance. The reason we have T cells, they evolved to protect us against viruses. And a virus is an infection of a cell. It could be, one in your lungs, and that would be flu. And sometimes it makes the molecule RNA, the virus, or other times it's DNA. But in either case, the T cells can sense that it has altered its DNA or RNA in the cell, and then the T cell will kill it. And that's how we get over the flu. It's how we get over chickenpox or mono, for instance, is the T cells kill those infected cells. And a tumor, we now know, is a mutation which changes the DNA in the patient's tumor cell. And T cells can sense that and can kill the tumor cells, because it's, in a way, very similar to an infection. The problem is usually the tumor cell remains very much like a normal cell from the immune system's point of view, and the T cell doesn't attack it vigorously enough. So that's where the CAR T technology comes into play. We can make very strong recognition or binding of the T cell to the tumor cell, and then it kills that without fail. I mean, that's what's really exciting. Now, so there are two approaches to solve the problem you brought up, because right now the big challenge to the field is, for instance, solid tumors and other tumors other than CD19 positive bone marrow tumors. And so I have several arguments why I am quite confident that's going to happen, where we will have successful engineered T-cell therapies for all tumors. So one is that there are many different antigens or molecules.
Can I say yay and then?
Yeah, but you have to understand, what I don't know is when. The time scale of this coming out is, and when it comes out, so I can say in mice right now, the principle's already proven. And but there are two kinds of T-cell therapy. There are the CAR T-cells that we've talked about, where it's an antibody fused into a T-cell. And then there are also T-cells given, and we've done this at the University of Pennsylvania, and there are companies doing this, where new T-cell receptors are put in. So T-cell receptors identify intracellular, the new proteins, the DNA mutations that I was mentioning, and they can tell inside the cell that it's been altered. And so they kill in the same way that a CAR T-cell does, but they use a different recognition system than the antibody system. And so we know that T-cells can kill solid tumors when they see these mutated antigens through the T-cell receptors. That's the basis of so-called TIL therapy, and TIL stands for tumor infiltrating lymphocyte, where lymphocyte really is a scientific name for T-cell. So TIL therapy has been used for a long time for metastatic melanoma, and metastatic melanoma is a tumor where chemotherapy has never been effective, and now we have, revolution in that. It's the first solid tumor that's where up to 50% of the patients now have long-term remissions from immunotherapy, a tumor completely refractory to chemotherapy. And it's been done with either so-called checkpoint therapies, where antibodies are given to prevent the immune system from shutting off this anti-tumor response. And it's also been done with TIL cell therapy, so that those are adoptively transferred. So the patient's own T cells taken out of a tumor, they're grown up in the lab, and then given back to the patient. Now, so what I think is that in every patient there will be combinations of targets that will be either the T cell receptor target or CAR targets. And there's one more nuance to understand. So right now we have a CAR T cell that is always on, if you will. It's like a light switch, and it's always on, and that CAR is always on the surface. And if the T cell runs into a cell that has that CD19 target, it basically shoots before asking. Okay, it's in military terms, it's just there is no question asked. It just kills. Now, it's possible using transistor logic, and engineers will know about so-called Boolean gates, the way transitions work. They can do logic, and the cell, for instance, it can ask, just as computers do on an initial logic questions, it can do and or not gating. So it's possible to make a CAR T cell so it can say, does the target have two targets, say target A and B? And if it has both of those targets, it kills. And you can find two targets on every tumor cell where no two targets like that are in normal cells. So then it can find, that's the really important aspect. On normal cells, they may have target A or target B, but you can always, with the technologies we have, you can identify unique combinations that are on the tumor cell and not on the any normal cell found in the body, and then they become tumor-specific killers. So that Boolean logic gating, and we're calling them, and there's some people call them smart cars, because they have to, but they can be rewired so that they only kill when there's a certain combination of targets on the tumor cell, and then that's going to have to be made so that it's specific and not on normal cells. So that principle's already been shown in petri dishes and in mice, and it needs to be worked out now in patients with tumors. But yeah, and the last part of that equation that makes me optimistic is so you could always ask, well, can T cells really kill solid tumors like pancreatic cancer and brain cancer, where there's been no real progress for decades and decades? And the answer is, if we make CAR T cells in the lab from a patient and take their tumor cells out from a biopsy, we can show in the petri dish that the CAR T cells can kill those tumor cells. So the issue is making them so the CAR T cells also don't kill the normal cells in the patient as well. And those are the approaches we just discussed.
Well, it sounds so exciting. It's certainly made me feel a lot better, that there's going to be more solutions to the end game than what we're confronted with right now if you have myeloma.
Yeah, it's, in the case of myeloma, it's the most common bone marrow malignancy in adults. And as you said, it's been really not curable. What's happened is we've got a series of targeted therapies now that are much better than the chemotherapy we had in the past. But myeloma has a relentless property where it seems to always come back. And it may be that there's something called a myeloma stem cell, which is resistant to all known forms of therapies, and then that's what leads to these inevitable recurrences. And so with the immune system, it may be possible to target both the myeloma stem cell as well as the common myeloma tumor cell that causes all the problems with myeloma. So I
that's
think
a good part of the shoot before you ask, isn't it?
Yeah, yep. So we need to target the myeloma stem cell as well as, the mature myeloma cell that causes all the bone damage.
Okay. One last question, and you had talked about the way that you use the HIV virus in order to introduce, this antigen. And we hear a lot about CRISPR, and that CRISPR is better than the existing systems, and that it's far more accurate. And my assumption is that there must be some benefits then to that as far as the cost and also your ability to make it do what you want it to do, and to not have as many, say, off-target issues. But could you explain, is CRISPR something that you will be using in the future, and not use the HIV, or how does that work into this whole program?
So we all, it's just an example of now how basic research has led now at the cusp of ways where we think we can actually cure diseases in a variety, even inherited diseases, which was unthinkable in the past. So this is an area, so-called genetic editing is the ability to change the code in DNA. Our DNA is about 3 times 10 to the 9th, or 3 billion different base pairs of this DNA, and And sometimes, a mistake in just one of those can lead to even lethal diseases, or can cause cancer. So you can have congenital, diseases that you inherit, or you can have acquired ones which happen, for instance, and lead to cancer through mutations after you're born. And so genetic editing offers the ability to repair these, and it can be done in two forms, in a basic terms, one being in the laboratory. If you can take the cells out, genetically edit them, and then return them. So that could be in bone marrow-derived cells like T cells. It happens to be that today the first ever in vivo gene editing was reported by a team led by Sangamo, and they used a gene editing technology called zinc finger nucleases, and they injected it into a patient who had a congenital disease in order to attempt to repair the gene. So it's too early to know if that's going to work, but in a laboratory in mice, it does. So when you actually look down in the different technologies, they alter DNA sequences, and we're fortunate to have a number of tools. The first one ever used in patients was the zinc finger nuclease, and we actually did that beginning in 2009 in patients with HIV, and we edited their T cells to make them so that the HIV virus could no longer infect their T cells. But that was done in a laboratory, much like our CAR T cell engineering. This is going to be complementary to and will not replace technologies like using lentiviral vectors or HIV to introduce genes. So there's one which is technology which can bring in new genes to the cell, and then there's another which can change target genes and to achieve a benefit in the patient, and they're really very complementary. And you mentioned the CRISPR technology, which uses an enzyme derived from bacteria called Cas9, which it'll cut in the DNA, and you can target it with amazing precision. It's truly the needle in the haystack. So of these 3 times 10 to the 9th or 3 billion different base pairs in our DNA, you can target which one you want to change with this CRISPR-Cas9 system. And its real advantage over the previous gene editing technologies, like zinc finger nucleases, is it's more rapid in the laboratory to test many different targets. And so it's very flexible, and I am quite certain that we're going to see many of these technologies used both for in the laboratory with, for instance, bone marrow cells are altered and then given back to the patients, or in certain tissues they'll be directly injected, and those areas will be modified as desired by systems like CRISPR-Cas9.
Okay, well, fantastic. And I now am totally convinced that you are a special person, and that from now on I'm going to start calling you Dr. Carl Einstein Jr.
No, just you have to understand. I mean, this is immunotherapy has been an idea around for more than 100 years, to try to treat cancers, and so the ideas are not new. I mean, there was a surgeon in New York City in the late 1880s and 1890s, injecting bacteria and products from bacteria into patients' tumors because he thought he could activate the immune system. And in fact, he did. Some patients were cured when they were deliberately infected with bacteria. And we now know in retrospect that was, it was activating the immune system, but it never became FDA approved because it was not reproducible enough, and it was also quite dangerous. So the idea to use the immune system has been an old one, but we didn't have initially the tools that were, good enough to make it work. And now we have, through all, years and years of very, basic research on the immune system and ways to modify genes. And in fact, in the entire human genome sequencing effort has had a major benefit in this cancer immunotherapy. So these old ideas now are able to actually be done now because we have better tools to get the job done.
Well, thank you. Jack, Aiello, are you on the line?
I am. This has been a great discussion. Dr. June, it's really nice to meet you over the phone. I've actually met you once before, a couple years ago at a Leukemia and Lymphoma Society meeting when you introduced Emily Whitehead and her parents.
Yeah. Thank you, Jack. Thanks for working with LLS. When you have an out-of-the-box therapy, one thing we've learned is that standard science often does not fund that research when it's out of the box. And I was unable to get initially, funding from our National Cancer Institute. They funded our development with the HIV virus that I described earlier. But when we wanted to do clinical trials in patients with leukemia, I was unable to get that funding. And it was only through the Leukemia and Lymphoma Society that we were able to start that trial.
That's what I understand. In the trial that you discussed, I know that you reconfigured the T cells to recognize the CD19, and yet I also think you gave that treatment to some myeloma patients, and I had heard that some of the myeloma patients responded, and yet I never understood why, because we don't really have CD19 as a marker on our myeloma cells, I don't think. But I was curious if that's true. Did they respond, and what's their condition today?
So I'm going to go back and describe what I think is this stem cell, that there's a cancer stem cell, and that myeloma cells may be, in a way, like a chameleon and change the molecules on their surface at different times during their lifespan. And so we have data, and there's a scientist at Johns Hopkins named William Matsui who's shown over the years that there are normal B cells that do have CD19 that appear to be the precursor to the mature myeloma cell. So we, as you mentioned, we treated the first patient on a trial just testing our CD19 CAR with in myeloma, and she had a complete remission, and she's doing very well now 4 years after treatment. And she had 10 different kinds of therapy before that had never worked. When we looked at her myeloma, only about 2% of the myeloma cells had CD19, and yet all our myeloma cells went away when we treated her with the CD19 CARs. And so we've now done a trial, and about 2 out of 10 patients had quite strong responses, again, to the CD19 CAR, but 80% of the patients didn't. And there's another molecule that we are testing, and other companies are now testing for myeloma, called BCMA, and BCMA stands for B-cell maturation antigen. And we're having responses quite routinely in patients using CARs that target BCMA. So this is what I'm going to describe now has not yet been published, but we took 10 consecutive patients with myeloma and tested their cells in a petri dish, and then put them into an assay to look, did we eradicate all the myeloma cells, even the long-term so-called stem cells. And when we treated with the CD19 CAR, it eradicated about 20% of the myeloma patients, where all the cells were killed, that had long-term, ability to cause myeloma. And with BCMA, it was about 70%. And when we gave a combination of CARs targeting CD19 and BCMA, we had 10 out of 10 eradications in the laboratory. So we're really excited about that, and we're just beginning a trial with no one yet treated, where we're going to give them the combination of CD19 and BCMA CARs. So if you will, it would be a cocktail of CARs. And that may become common, I think, where many tumors will be treated with a combination in order to eradicate every single last tumor cell.
So that was actually my next question, and that is it possible to build a CAR T to recognize more than one antigen? But maybe through the logic, or maybe just through having separate CAR T development, you're able to do that.
Yeah, it's a very exciting area of research and there are a number of ways technically to do this. I was mentioning this Boolean logic, where you could have a CAR target A or B or C, for instance, maybe 3 different CARs in the same T cell. So we now have technology to do that. And there's a trial that just opened up at Stanford targeting 2 molecules for leukemia. It's targeting CD19 and at the same time CD22. And so I think we'll have the same thing in myeloma. I mean, as I just described, but using BCMA and CD19. And I think we're just at the surface. We're just scratching the surface of, next generation therapies that will be both more potent, have higher cure rates, and then less toxic.
And then I also hear in the future that this technology, CAR T technology, might be off the shelf or might be replaced by something called BiTE technology. Can you share a little bit about that?
Yeah, so those are 2 different concepts. So off the shelf, I mentioned that, CAR cell therapy really is a form of transfusion medicine. And instead of red cells or platelets, which are routinely transfused, this is T cells. And an off-the-shelf CAR T cell would be where we could, where it wouldn't be the patient's own T cells. And if that could happen, it would make the process much simpler. Because right now it's a logistically intense effort where the manufacturing process is unlike anything in the pharmaceutical industry, really, where it starts with a patient. They donate the T cells in a process called where the blood is taken out by either just blood donation or apheresis machine. The cells are then shipped to the manufacturing plant, and then about 2 weeks later they're returned back. They're now frozen, and then the cells, CAR T cells, then are given to the patient by about a 5-minute infusion. So the cells are just thawed in a water bath that is at the same temperature, a body temperature, so 98.6 Fahrenheit. When the cells are all thawed out, the CAR T cells are just infused over a period of about 5 minutes. So that's how the current FDA-approved CAR T cells are given to patients. An off-the-shelf one would be more like a standard pharmaceutical product where it could be made in large batches. And what it requires then is that we have something like we do in the blood cell transfusion where we can have universal donors. So it happens to be that I'm an O-negative donor, and I can donate blood to anyone because of that. About 15% of patients have this the blood type of O-negative. We don't have that for T cells unless you have an identical twin. So if you're an identical twin, you're lucky you got someone else who can be an off-the-shelf donor for you. But for most people, that's not true, and we need to be able to genetically modify the donor T cells so that they don't do 2 things. So one is, I mentioned with bone marrow transplants, you get graft versus host disease. That's where the incoming bone marrow attacks the patient. And T cells are what do that. So if I would take someone off the street, make CAR T cells, and infuse them in a patient, the patient would later get graft versus host disease from the infused CAR T cells. So we have to remove that with genetic editing, the ability of the off-the-shelf cells to cause that graft versus host disease. And then the other problem happens is that the patient themselves will reject the incoming CAR T cells, and that's called host versus graft. So it's the other way around, the patient's immune system. And so then these off-the-shelf T cells need to also not have that happen. There are trials now testing these off-the-shelf cells, and very early stage trials are being done in London at the Great Ormond Street Hospital for infants with leukemia. And in New York City, there's a collaboration between two biotechs and pharma, Selectus, Servier, and Pfizer are working on making off-the-shelf T cells for patients with leukemia. So I think in the end we'll have, standard oncology has had what we call induction chemotherapy and then maintenance therapy oftentimes. And I think we may well have two kinds of CAR T cells in the future. One that's the kind that can last for the rest of your life in your body, and that would be your own cells manufactured. And there will probably be, I think, an off-the-shelf form of CAR T cells that could be at any community hospital and then given right away, and then later probably for long-term. I don't think the off-the-shelf T cells are likely to last lifelong in a patient, unlike when you use your own T cells. So I think we'll have both forms of induction and consolidation with these kinds of engineered T cells.
Thank you, Jack. We're just running a bit over time here. So, Mike, you're on air. Please ask your questions, and then we can just open it up for the audience.
Hi, Dr. June. I'm not going to ask you any questions because I want to make sure we have time for people who are on the phone to ask you questions. But I did want to tell you a very brief story. In 1973, I met a man called Barry Marshall on the way to a meeting in Belgium, just after he had shown that a bacteria caused ulcers.
Uh-huh.
I published that paper. I told him at the time he would get a Nobel Prize. And I'm telling you now, I hope you and your team get a Nobel Prize for this work.
Well, thank you. I did notice.
I know how rare it is that something like this happens.
Yeah, well, I've been very blessed. I have an amazing team that I've worked with for, many of the people I'm working with, I've been working with for more than 20 years. But I noticed you work, in prostate cancer.
Right.
And I wanted just to say that we have just treated our second patient for patients who have metastatic hormone-resistant prostate cancer with CAR T cells.
Excellent.
Yeah, it's a trial sponsored by the Prostate Cancer Foundation, and so it's a very early stage, pilot trial to understand if we have a safe target and if we can make any headway in these patients right now who have no options.
Right. I did hear a presentation you gave a few years ago at the Prostate Cancer Foundation meeting in Washington, so I have been optimistic ever since.
Yeah, so it's finally happening. So we're excited about that, but we just don't know how long that road's going to be in patients with solid tumors. It's a complex problem. Okay. Well,
but
thank you.
I really want to give people on the line the chance to ask questions, so I will waive my right to ask any others.
Okay.
Thank you so much, Mike.